There we go. Always nice to hear that. Of course, before we go any further, myself and Kaylin are joining you from Victoria, BC, Canada. The videos you were watching were from Ocean Networks Canada's many excursions, and we invite you, of course, to check out their website, but we have to, and we want to acknowledge with respect the Lekwungen peoples on whose traditional territory the university stands, and the Songhees, Esquimalt, and with Saanich peoples whose historical relationship continue to the land to this day, continue with the land to this day. Like I said, the video was brought to you by Ocean Networks Canada. We manage cable observatories along the Canadian coastline and collect data for scientific research to help communities, governments, and industry make informed decisions. This data is freely available to anybody who wants it, citizens, scientists, students, and enthusiasts. So please don't forget to go to oceannetworks.ca. It's available 24 hours a day, 7 days a week, even when the expedition wraps up. As mentioned, my name is Monica Peltz. I am the K-12 education coordinator with Ocean Networks Canada, and I have the pleasure of introducing or letting my co-host introduce herself.
Thanks, Monica. I'm Kaylin. Welcome, everyone. Thank you so much for joining. I'm the Ocean Literacy and Events Administrator.
Excellent. And of course, we couldn't do this without our partners. So a big thank you to everyone who made this possible today. Obviously, Fisheries and Oceans Canada, woohoo, Council of Haida Nation, New Chalmers Tribal Council, and of course, ONC. So thank you to all our partners.
Yes, and this expedition exploring and documenting the diversity of these deep sea ecosystems is an endorsed activity of the UN Decade of Ocean Science for Sustainable Development. And Challenger 150 is a global cooperative focused on deep sea biological research, formally endorsed as an Ocean Decade program.
Alrighty, I will let the ship tell us most of what's going on, but just a quick recap, get you thinking of those questions. Some of the goals of this expedition include mapping and naming uncharted areas, exploring life on newly discovered seamounts, searching for cold seeps and their chemostatic communities, confirming the existence, activity, and community type of little-known hydrothermal vent fields, documenting the oceanography around deep sea ecosystems, documenting marine mammals, seabirds, and transient species, and of course, this right here, what we're doing right now, science outreach, communication, and engagement. And this is really, we really mean that. We want to hear your questions, we want to engage with you, so we are very excited that everybody is here.
Owing to the region's uniquely small, active, and nearshore tectonic plates, there's an impressive concentration of seamounts, hydrothermal vents, and cold seeps that are globally rare biodiversity hotspots. And we're going to let the ship tell us more about that. While the main goal is conservation through protection and recovery, the expedition partners recognize the importance of their research in ensuring healthy oceans for all. So you've met myself and Monica, and now we will head over to the ship and let our wonderful crew introduce themselves. Yes, and a little reminder to pop your questions into the Q&A. As of now, you can begin doing that and we will get to all of your questions. Alrighty, to the ship.
Okay, sangay la dalang wadlawan skiljada hinudi kyang hade wadlawan ne sao shangulang. So good day, everybody. My name's Skiljada. I work for the Council of Haida Nation. I'm the marine biologist / marine planner. I've been helping out a lot with the collections on the ship, really just with everything that we've been doing, which you'll hear more about. But first, we do just want to thank all of our partners for making all this work happen. DFO, CHN, NTC, ONC, all our fun acronyms, as well as the Canadian Coast Guard. It's really a lot of hands make lighter work kind of situation, like a lot of work and effort goes into this, and being able to see all these special places is a real privilege. You know, we have multiple nations working towards protecting our oceans, co-governing important marine protected areas or areas of interest to become marine protected areas. And in order to protect the ocean, we have to understand the ocean. And the best way to monitor deep sea ecosystems is using very fancy remote technology taking water samples, taking specimen collections, which we'll tell you more about. So how for joining us today. Thanks, Gildada. So hi everyone, my name is Tammy Norgard and I am the program lead for the Integrated Conservation Monitoring Program. And what I do is help to organize these large multi-partner surveys. And so we are here on the west coast of, we've pretty much covered the whole west coast of Pacific coast of Canada here. We are, that's right, Gildada's got us, we are right here right now. And we've been able to travel up the whole coast and around, and we're doing a loop and we're on our way back home now. We've got a few more days left. Right now we're on top of Union Seamount, and you'll hear more about what we're doing at this very moment from more people from us. But as I would like to show you where we actually are. So we're way out in the middle of the ocean. This is a drone footage of our ship out here. We're up in the clouds. It's a very large ship. We have over 40 people on it. We are conducting over, actually, I've never counted how many different research projects, but probably at least 10 different research projects. And we have a team of 15 scientists working together to make it all happen. So you're going to get to meet all of these other scientists and how everything, and hear about the different projects and different equipment and programs we have running. So, oh, and here's a nice close up of our ship. And on top of it, what's really important, I would like you guys all to notice this. This is a 70-meter long ship. And right now we have our multi-beam pole in the water, and this is what it looks like. Right now the ship below us has this pole down and it's mapping the bottom of Union Seamount. And this golf ball on the top is how we're all talking to you. So we're reaching you through satellite to all of your homes today. And it's back to you.
So 2 days ago, I believe it was, I was lucky enough to be able to be the scientific dive leader for on Kinkless to the peak, to the pinnacle, which was an amazing experience. And the life that we saw there was so abundant. We had gigantic ancient corals. There was so many rockfish, which we'll hear more about and see more footage of. There was all kinds of, there was whale bones as well. That was a fun find. There was invertebrates like echinoderms, like sea stars, prowfish. It was really special to see that it was a refuge for critically endangered species. So there was tons of juvenile rockfish. So it was a nursery area, and there was a lot of pregnant yellow eye, which is an endangered species and a type of rockfish. And there's also Pycnopodia, those big sun stars. And those are critically endangered after the sea star wasting disease. And we saw a lot of healthy Pycnopodia species, which was really great news to show that the marine protection of this area is so important, because the life was just bursting and the geology was also phenomenal. There was like lava cliffs and ancient beaches, like sandy bottom with like pieces of clams and shells and all kinds of things. It was really phenomenal. And yeah, we'll hear more about it, and please ask lots of questions about it, because it was the most exciting dive that we've done so far. But tomorrow we're also doing hydrothermal vents, so that'll be cool too. So tune in for that. So now I'll pass it out to our crew on the deck. We'll see Charisse tell us about the ROV that gives us these cool images.
Hi everyone. Hi show team. Hi people on the internet. My name is Dr. Charisse Dupree, and I am the lead deep sea researcher on this expedition. So I work with everyone on board, but I work with people who aren't on board too. We have national and international collaborators as far away as Germany. And a lot of us are here using, or the scientists are involved, using the PlagixROV. So we're not just scientists on board. There's a team of technicians and pilots, and it's all to run this robot right here. So this is the PlagixROV remotely operated vehicle, and I'm going to be the first team member to show you a little bit about what we do, and we'll have the rest of them show you afterwards what they do. But I'm going to talk about the ROV. I think I got the neatest thing to introduce. So I'm going to show you some of its components so you can see just how useful it is to have a robot submersible when you're a deep sea researcher like me. So I'm not sure if you can see it, but here we got the big yellow floater pack. It's high density foam. There's a lot of metal and computers on board this vehicle. This vehicle leaves the ship and is tethered to it. So there's a cable that feeds the robot power, relays information, and it's also how we get imagery and information back from the vehicle. Inside here, tucked away, you might not be able to see it, but there's a big silver mechanism in there. That's the brains of the ROV. It's where all the power and information comes from. And then there's a whole bunch of modular systems on it. Probably one of the most important systems is the driving system. So all around the vehicle are thrusters like this, pointed in different directions, that the pilots on board the ship can control. And so we can fly the ROV around the seafloor. This vehicle goes to about 6 kilometers into the deep sea. Actually, that tops out our Canadian Pacific Ocean. Ours kind of stops at a hard 3,000 meters. So this vehicle can go a lot deeper than our oceans are even deep. So another neat component of this ROV is that it's got collection capability. So I'm going to show you down here are sample jars, and they're connected to this what looks like a vacuum cleaner hose, and at the end here it's connected to an arm. And see, so we can maneuver this around the sea floor and collect very delicate items off the sea floor and put them on the ROV, and when the ROV returns to the ship, the taxonomist scientists can look at those. Other things that you can see here, we've got another arm on this side. The main cameras are in the front here, so this is the front of the vehicle. If I was a deep sea creature, the vehicle would be looking at me. Another component of the vehicle that is very important is its lighting system. So it's got banks of lights all over it, and that's because if it didn't, we'd just see what the deep sea is naturally like, and that is pitch dark. So there is no light that gets down to these environments that we're mainly diving to. And so we introduce that light so that we can just flashlight around, non-invasively see these animals, document them in their natural habitats, selectively sample a few, but otherwise leave them be. Also point out here, this is a marker with a weight on it. So at some select sites, we'll drop off these markers, and this vehicle has an amazing navigation system on it, and we can relocate those years later to check out how the environment is doing, how the conditions have changed, how the animals have changed. We also have downward facing cameras. We've got other video cameras. We have push cores. We got a bio box. We have a scoop for collecting animals. We have strobe lights. So again, an amazing piece of equipment that allows deep sea researchers like me on the ship, our partners abroad, to watch in real time what this vehicle is doing and help us coordinate this large research effort. And I'll just pull in one of the pilots. This is Jesse from Pelagix. Jesse pilots the ROV and works very closely with the scientists. Jesse, is there anything that I missed about the ROV that you think the people at home should know?
I think you covered the important things. The rest gets a little more technical and may not be clear.
Okay, well, we'll leave it. So Jesse said I covered the majority of it, but the rest of it gets very, very technical, and he suggested that we leave it there because that's exactly like the scientists like to know. I don't really want to know how it works, just can you make it work for me? So thank you to Plagix, and with that, I'll pass it back inside to the rest of the team so they can tell you about the tools that they use and the environments they've explored and the experiences they've had so far. Thanks.
Hi everyone, I am Lindsay Clark. I'm a graduate student with the University of Victoria. On the screen here beside me is the control van. This is where our pilots from Pelagix sit and operate the ROV when it's down in the water column. And while they're doing that, I'm inside the lab annotating everything we're seeing and everything we're doing, like putting down those markers that Therese mentioned. Beyond the stuff that's happening down below the waves, I'm also annotating and taking note of all the wildlife we're seeing on top of the waves, including probably my favorite one so far, the Blackfooted Albatross. So These birds they're huge, they're massive. Their wingspan reaches up to 7 feet, and they can live 40 to 50 years. They spend most of their life on top of the waves or flying above it, and they only ever come to land to breed. They do this once every 1 to 2 years, and they only ever have one chick. These birds mate for life, and yeah, they're pretty amazing to see surfing the waves.
Can you remind us, what do you mean by annotate? We have a very diverse audience today, so maybe just a little clarification. What's an annotation?
Yeah, thank you. That's perfect. So annotating, I'm just writing notes. I'm taking a whole lot of notes on the computer. And we share these annotations, these notes on the website, so that when we're transiting or when we're diving, you can see and take note of what we're seeing, and we're sharing what we're observing with you.
Thank you.
Hello, I am Tammy Gomez. I operate all the video gear that gets the streams out to the world. If you ever watch our live stream, that's what you get to see, as well as recording all the video from this main camera here on the ROV. This is what you see. This is the main eyes of the ROV. There's numerous cameras around it, but this is our main one. It is 4K camera. And on top of recording all the video for later use, I recorded several different codecs or different formats, so you can use it on social media right away, or to be able to send it around to different scientists all over, so then they can later look at it and annotate it as well, or log and record observed data. But also, when we're not at sea, we tend to find trash or different things, or not on a dive, I should say. We find trash and we'll pick it up, but sometimes that trash turns into treasure. So this is a glass ball that we found. It was covered in these little barnacles, and we have scraped some of them off. We're gonna do some biological sampling and different things with it. So that treasure can also be good use for the boat. We're actually having a raffle now, so who gets to go home with this nice piece of treasure.
Tammy, I want my name in the raffle. Can you tell us, where did that piece of treasure originate? Because I think that's pretty significant.
Japan.
Yeah,
That's what they're telling me.
I believe so. I believe that's a Japanese fishing float. So yeah, throw my name in the raffle there too. That is a big prize out at sea.
$1 a ticket. Taking bets. Put it in the queue. No. Carry my treasure out of here.
Oh, here's a short video of us retrieving it earlier. So that Shree's carrying it in. And yeah, cool. Okay, hi everyone. My name is Shelton Dupree. I'm on board this expedition as a contractor, a cameraman and filmmaker, and I'm here to document all the science that's taking place on board. Sorry, there you go. And so yeah, so my job on board is to shoot everything that's taking place, photos and videos. And the way I like to say it is bridge the gap between the science that's happening and the general public. So creating, you know, photos for people to post and share and tell the story, and then creating a final wrap-up video at the end to kind of summarize everything that we've done on board. So one of the things that I'm going to talk to you a little bit about today is some of the specimens that we've collected. And what you see behind me right here is what's commonly known as a Seapig. And what we have here in this jar is the Seapig itself. So I'm going to come up, I'll bring it up to the camera as best as possible so that you guys can have a look at it. So that is the Seapig that you see on the screen behind me. We brought it up from, I think it was about what, 2,000 meters. So that is the Seapig. And so part of what we do is we collect these samples. So we get to see them. We get to see them in nature. We get to take video of them like this. And then we get to bring them on board, and we have an amazing photo ID station set up. So we bring all the specimens in and get good photo IDs of them so we can properly identify exactly what they are. So here's another example. This was a little squat lobster that we had. This guy is about this big, maybe an inch long. And here we have a little shrimp, beautiful brittle star. And yeah, so that's a bit about the specimen collection.
Hi, I'm Chelsea Stanley, and I am an oceanographic technician with DFO, and I'm on board as the lead oceanographer. So a lot of this trip is obviously focused on what is on the bottom of the ocean and on these seamounts, but a super important part of the story is the properties of the water that surround the seamounts. It highly impacts all of these animals, so it's important for us to understand the different water qualities that are in the water surrounding the seamounts, mostly the chemistry. So we use this tool, it's called a rosette. We launch it off the boat. It has got an electronic sampler here called a CTD. And so that's electronically sampling the conductivity or the salinity, the temperature, the depth, the oxygen concentration, the fluorescence, and a few other chemical properties that are important for us to understand. This can go down very far. I've had it so far down to 2,000 meters. And then on board this rosette, swiping the TV instead of the iPad, we have these 24 10-liter bottles. And so these bottles are remotely closed. So from the ship, I can send this vessel down to 2,000 meters. I can close a bottle, and then the water that comes up will be from that 2,000 meter depth. From that, we sample these into different kind of containers, glass vials, and we then do more work that can take the chemistry of this and figure out the oxygen concentration, the salinity, nutrients. We're looking for eDNA to see what kind of animal's DNA may be in the water surrounding the seamounts. And then from that analysis, we're also able to ground truth and just make sure that the readings that are coming from the CTD are correct. Another role I'm playing on board the vessel is collecting some hydroacoustic information. So we have a couple different types of hydroacoustic tools on here. We have a multi-beam sounder, which you'll hear about later, and then we have a split beam sounder. The cool thing about the split beam sounder is that we actually can see in the water column different types of biological groupings. So we'll see fish schools, swarms of krill, different types of layering in the water column. So one thing we saw when we were at Skon Kingless Bowie was we saw huge schools of rockfish. So this is the sounder. I hope you can see this okay. This bright red was the bottom. It's moving fast. And then we could see a huge swarm of fish on the acoustic sounder. And then when we panned up with the ROV, we could see all of these hundreds of rockfish above us. There we go. Bottom, swarm of fish here. And then the next slide will show you a better idea of how many rockfish were above us in the water column, which we could see with the sounder. So that's ground truthing what we were seeing on the sounder using the ROV. So just another tool to show us what's in the water column. On to bongos.
All right. Hi, everyone. So my name is Daniel LeBay. I am a graduate student at the University of Victoria, and I'm here also helping out with the oceanography side of the cruise. So what we have here is a set of bongo nets. So these are the nets that we use to sample zooplankton in the ocean. So that's all the little organisms and animals that float around in the water column. How these nets work is we'll lower them down to a certain depth, normally about 250 meters, drag them back up towards the surface, and all the animals that are too slow to swim away will actually get funneled into the end of these nets and into something like this, which we call a cod end. Then we're able to take the cod end off, take it into the lab, and do all sorts of analysis. Typically, half of the one side of the cod end goes towards preservation, so we can preserve them and see what species are present in the water column. Then the other side goes for freezing, where we can see the biomass, so how much the zooplankton weigh in that water column, and also a whole other sorts of tests and analysis to see what they're made of and how good of a food they might be for the rest of the ecosystem. So studying zooplankton is very important for particularly to figure out how much food is actually available for all the fish species, because the majority of fish do end up eating some sort of zooplankton. And yeah, so here we actually have a little video of the Bongo net moving through the water column back up to the surface. If you look really closely here, you can see a little fan going, and that's actually the flow meter. So that's what measures exactly how much water is passing through the nets. So we know we're able to quantify how much zooplankton we catch. And typically I did say that we go down to about 250 meters, but sometimes we'll go even deeper for a deep Bongo net. One time we went down to about 1775 meters, which is almost 2 kilometers below us. And when we brought the net back up, then we get some different critters. So one thing that we got was right here, which is a deep sea jelly. These organisms are very cool. They float around in the water column, typically below 900 meters, but are sometimes seen on the surface towards higher latitudes in the Arctic. And when they float around, they kind of hold their tentacles over here. They hold those up over their heads and wait around waiting for copepods or small crustaceans to capture and eat. And that's all I've got to say. Here's Georgia.
Hello, my name is Georgia Clyde. I'm a physical scientist and geographer. I work for Department of Fisheries and Oceans, and I'm going to talk to you about the mapping that we've been doing. So mapping has been a huge part of this expedition, and we've been mapping at 2 very different spatial scales. One at sort of a whole seamount scale, and the other is a very detailed, some 10 meter by 10 meter area. I'm going to show you both of those. But first up, this is the multi-beam unit that we are using for collecting bathymetry data of our seamounts. And this is actually being led by Chelsea, who you just met a moment ago. And this is a DFO initiative. And so with this tool, just see it getting into the water here, it gets lowered down off the side of the boat there. And then we drive along and it collects data as we move. And this really took a village to get going. So we're happy that it's working. But this is the output from the multi-beam. And so Chelsea and the crew last night were sort of pulling an all-nighter, mapping Union seamounts. So you're actually seeing here, this is today, what we've been able to do. And Union, despite being the 2nd largest Canadian seamount, was really unmapped until this very moment. We're actually finishing data collection as I talk to you. The last 30 minutes is happening right now. And so this is really cool to be able to do. And I think our last count was that we have confirmed 8 seamounts, mapped 8 seamounts, discovered 4 new ones that we didn't know were there. So this has been a huge component of our expedition. And the other thing that I alluded to earlier was this really detailed mapping. So this is the work that I've actually been involved in. And so we take imagery and video from the ROV, and I use a technique called photogrammetry to build 2D and 3D models of these really small study sites where we've deployed these survey markers. I think Sharice showed you out on the ROV, those markers that we drop down. So we revisit these sites, and this is a 3D model from 2018 of a cliff. Let's see if I can get it to play again here for you. So if we zoom in, we're facing the cliff face here. We'll pan along. You can see the striations in the rock there. And then there's a big coral up here on the right. And so 2 days ago, we actually went back to this site and we collected the data. And so I will be looking at that as soon as we get off the ship here. But just really cool to be able to see these reconstructions. And then I think I have a cliff or a clip of the cliff, just a little bit over, where we can see these lava flows, the striations in the cliff face. So this was a highlight for me. The geology here was just so amazing, and hopefully we can capture that again in our models. So I'm going to pass you off.
Awesome. Hello, Georgia. And since we're here talking about on kinkless again, I realized I forgot to show off my cool props. So we have a really large rock from the eastern side of on Kinkless. There's a lot of holes in it because it was an explosive volcano. and the gas bubbles get trapped. And as soon as it hits the cool water, those bubbles leave these big holes, which are crevices for lots of critters to live in. And from the top part of San Kingflis, when it was an island, when it was above water, it was a sandy beach. It eroded those volcanic rocks into small sediment like beach sand. So I'll show that here. Hopefully it can focus, maybe not. Either way, you can see the dark color is the rocks and the light color is shell pieces. So there's pieces of sea urchins and corals and clams and snails and things. So yeah, I hope you get a good sense of all the different things that we do in order to learn more about our oceans. So for that, I think we will pass it back to our hosts, and we're all happy to answer any questions.
Why don't we jump in actually to a really great question, because you kind of maybe touched on it, but we had someone ask, are you doing any benthic in-faunal sampling, which I'm interpreting as, are you taking any geological samples? Would you describe the samples you just showed us as benthic?
Yeah, they're from the bottom. We don't collect rocks intentionally. We're more focused on specific animals that we need to know more information about. So I believe we collected that rock when it had a sponge attached to it, or it was a small coral. I can't remember which. So it was kind of like a bonus find. And, you know, sometimes it can be taken as souvenirs, but everyone kind of agreed that a piece of San Kingflis could be gifted to the Haida Nation, or like in the Haida Nation's hands in order to use it for outreach and that kind of thing. So that's really special. It's a lot more than just a souvenir. It can be used for really important things. Yeah.
Wonderful, thank you.
Hey, Monica.
So, hello, Cherise, welcome. Great, you made it in from inside. From outside, in from outside. There we go.
Outside. Yeah. I made the journey. And,
excellent.
The question was covered in fauna, right?
Yes. Yeah.
Yes. Yeah. Well, we're super excited because tomorrow we're going to do something completely different. So far, the survey has really been about seamounts. And tomorrow we're going to shift it up and we're going to go to hydrothermal vents. And so for the first time, we're going to outfit the ROV with push cores. And those push cores are going to allow us to sample the animals that we call infauna. They're animals that live in the sediment around the hydrothermal vents. So we're really excited. And today we're working with Merlin, the lead of the taxonomy, to make sure that we're all set up for tomorrow, because we haven't actually collected any yet. And again, we're super excited. And just a plug for tomorrow, we're streaming live the entire time. So if you want to come and discover these events with us, our dive starts at 7 a.m. tomorrow morning. You know, it takes us a couple hours to commute 3 kilometers into the deep sea, but then we'll be diving at these vents all day. And infauna is one of the things that we'll be checking out.
Well, thank you, actually, Leslie. And again, this really is live, folks. We are actually seeing your remarks. Leslie said, nope, not rocks. I mean those tiny organisms. So I'm glad you're here, Cherise, to put me on the straight and narrow.
Yeah,
I learn something every time I do this with folks, including my new word of the day, the infaunal. I'm going to get I'm going to sneak that into a conversation somewhere. We actually had 2 questions about the ROV that I'm going to throw at you, because I think you might be our expert. So the first one came in.
I'm dressed like the expert today.
Perfect. The first one came in a little while ago. And it says, I'm used to seeing 2 ROVs work in tandem to compensate for sea movement. Your ROV works solo. How does it compensate for the various forces? So how do you see things?
Wow. Really wish we had a tech in here that wasn't pretending and was actually a scientist. Okay, well, I know that we spend a lot of time at the beginning of the dive putting floats onto a very heavy cable, and those floats make it so that the cable is I'm going to say buoyant behind the ROV and gives it the ability to move around in the absence of pull from the ship. So I'm very familiar with two vehicle systems and or anchored systems where there's a weight and then the vehicle almost has a leash around it. And so we don't have that. And so I know that these floats that we connect to the cable that start really close to the vehicle, and there's many of them and they space out, and then it's just the free floating cable after that. And those floats are what allows us to compensate for any pull. But the ROV team has a special person who's in charge of navigation, who speaks in real time to the bridge, to the captain or whoever's driving the boat at that time, and they have to do every move together. So we have a little bit of leeway. We can kind of move, you know, tens of meters away. But if we want to do a big move like a transit or go to a new site, we are speaking in real time, the scientists to the pilots to the bridge, and everyone's moving together. So we hashtag it all the time, but teamwork makes the dream work. So my answer is the floaties and teamwork.
Those are dynamite answers. So one more question about the ROV. Is there a contingency plan if the ROV encounters an overly curious megafauna, for example, a whale on a dive? Now, I know ONC has footage of when we've run into an overly curious squid, but what happens if you run into an animal that's just as curious as we are?
Oh, I actually asked this on my first day out here, and what I got was laughter from everybody, because apparently the ROV is a formidable animal itself, and that like it's robust and it's you know, it's sturdy, and so I don't think that they're very concerned about being attacked. But I will say that when curious animals do come in for a closer look, they tend to block the cameras, and so I would think that moving away from the animal so we could see. But I'm going to pass it off to Tammy G, who may have insight into this more so than I. So here, Tammy G.
Hello. Yeah, so usually if it does, you get curious animals every now and then, but you just kind of have to sit and let them do whatever. The ROV is quite strong. They're not going to do much damage or anything, but it does create a good story. But in general, the ROV is actually quite loud underwater, and we have these really big bright lights also. So in the deep ocean where it's dark, most of the time we scare them away, or the vibrations may cause curiosity of maybe a skate, but as soon as they bump into something, they'll just scatter away. So we don't have to worry too much. The biggest thing is, depending on what you're working on, maybe the animal will interfere and you have to wait for them to move or kind of shoo them away or something, just like anything else. Other than that, we had the lasers. This particular ROV has some very powerful lasers. Those are 2 green dots you'll see on the screen that are about 10 centimeters apart, and we use it to help measure how big or small an animal is. And so you'll hear us say a lot, let's paint the animal or paint the coral or the organism, and that's just so we can get scale to see how big, because everything is going to look very, very big to us, but then when we bring it up, it's much smaller. So this gives us kind of some perspective. But the other day we had some rockfish, just like a cat following a laser. We had some rockfish following the lasers and trying to eat them, so we had some fun with that.
That's awesome. We've got a few questions about some more species. So what are sea stars and what kind have you seen?
Anyone going? I'm a biologist, but not a active biologist.
Okay, so I mentioned those Pycnopodia, those big sun stars. There was a different species of sun stars. I forget the name of the species, but they look really similar to those Pycnopodia, those big ones. Maybe we could even go back to the video, play it on a loop or something. And we've also seen these brittle stars. So these are all echinoderms. So they're related to sea urchins and sand dollars and sea cucumbers. Yeah, they usually have radial symmetry, so they look kind of circular. Sea cucumbers, sea pigs, they kind of break that mold. But yeah, there was a ton of brittle stars, which are a type of sea star. They kind of clambered all over everything. It's on Kinkless, and I forget what species that was, but just lots of diversity of different types of sea stars.
Yeah, big ones, small ones, ones with less than 4 arms, ones with like a dozen arms. Yeah, the diversity is pretty mind blowing.
Wow, very
Wants
cool.
to jump in?
Thank you, Skiljata. Also, another question, where did you find the Seapig?
Ooh, we found the Seapig on SUP 5494, which is the seamount that's very close to Haida Gwaii. I don't know if we can see it on here. Yeah, so it's really close to home for me. It's this little triangle beside Haida Gwaii. So it's the only seamount in the northern shelf bioregion. So it's not actually in super deep water, it's on the continental shelf. But it's still a seamount because it's bigger than 1,000 meters. I forget exactly the depth and the height. But it was really cool to explore this because it's never been dove before. So we didn't quite know what to expect. It was kind of muddy, which was unusual. Usually seamounts are a bit rockier. And yeah, that was where we found the Seapig, and we were all kind of oohing and awing because it was weird and somehow adorable.
Definitely a lot smaller than I thought it was going to be. I was expecting, you know, a pig size. We have actually a question maybe for you and maybe for someone else about the 3D reconstruction. What accuracy are you getting, and can you see coral growth between visits? Now, I know that when I was listening the other day, there was some discussion about whether or not corals were getting bigger or smaller. How can you tell if they've grown?
I'll pass it straight to Georgia.
Hi, great question. So this will be our third year of collections. Is that correct? For some of the sites?
Goodness. No, these are the second
Second,
time
yeah.
we've done.
So we're going to build the models for the second time and then see if we're actually able to measure growth and things like that. So stay tuned on that. But in terms of accuracy, you know, we're getting down to millimeter pixel size in some of these areas, and definitely seeing lots of detail on corals and things like that. So stay tuned to see what we can measure, but we're definitely going to be able to monitor a number of individuals and changes throughout the scene like that. I think Charisse wants to jump in here and talk about the biology a little bit. I could talk about the millimeter accuracy.
Like creeping
Yeah,
into
she's
the
creeping.
scene, and then I just push her out. No, Georgia really is our expert when it comes to the reconstruction, but just anecdotally from what we observed during the dives, we saw a lot of the corals were persisting in the exact same spot, because Georgia and I were sitting out in the van, and we had the models and the photos that she's already created from 2018, so from 4 years ago, and we were looking at that to help the pilots fly the exact same lines. And what we were able to see is so much of it was exactly the same. And then we'd come across one of the corals, and it was about 1.5 meters in size, and 4 years ago it was perfectly healthy. It was a Prinoa pacifica coral, and now this entire coral has no living tissue on it whatsoever, but there was a sea star thought to predate on corals sitting at its base. And so here, a 1.5 size coral potentially completely consumed by this predatory sea star. So it's amazing to see how things haven't changed, and then to come across something that's changed absolutely, and now only the skeleton of the coral is remaining. But yeah, I am surprised all over again when I remember that we're mapping these habitats at a sub-centimeter level, and it is just, that is so cool.
That is awesome. Just a friendly reminder to everyone, you can put your questions in the Q&A. I will be honest with you, the chat moves fast. So if I miss your question because it's in the chat, my sincerest apologies. But please do throw things in the Q&A and we will get to them as best we can. Kaylin, I know you have a question specifically for Tammy G. So we're going to have to pass this to Tammy G.
That's right.
Tammy G. Here you go.
Oh boy.
Tammy G. So the question is, are you an ROV pilot as well as a videographer?
Oh, I like to dream I am. I am not. I am just operate all the video equipment, and it's racks and racks of monitors and buttons, and I get to push all the pretty buttons, I guess. So I don't do much of anything directly with the ROV at all. Maybe sending some cables out or troubleshooting some networks or different things between the ROV control van, we call it, which you remember you saw where all the other screens. I have my own little setup of that kind of miniature version in the lab, and I monitor all that as well as monitoring the records, intercoms. So during the live stream, you're able to hear us. So I have to monitor everyone's volume, when they get excited or sleepy. I have to turn them up or turn them down.
Awesome. Okay.
We don't get sleepy often on these. They're pretty good expeditions. Good
Oh, that's good to hear. We've got another question maybe you can answer. How do we access the live stream, and can we look back at that adorable Seapig?
Oh, I think there will be highlights coming later on. I'm not sure. C2? Oh, I'm being told it's already on C2, which is part of Ocean Networks Canada's website, as well as if you just go to Ocean Networks Canada, maybe Monica can put that in the comments or something, the link to that, to be able to just go ahead, click on it, and you'll be able to see the live stream. We stream 24 hours a day. Sorry to say at night, it's pretty dark. You don't maybe see a light or something, but during the day, even if it's deck ops or nothing's going on, you'll still see the deck. You'll see us walking around. So don't be afraid to say anything. Or we did a nice little dance with pool noodles earlier today. So that's what they're all imitating. Oh, so you can actually go, all annotation is visible underneath the video. So you can actually go on there and search for Seapig, and they'll be able to help you pop up that highlight or that video or any clip you particularly might want. So they could search for us dancing with the yes, we did. There's also Slido, right? That's what it's called. There's also, you can send messages 24 hours on Slido, and even if it's just to say hi or tell us where you're from, we enjoy getting the messages. We can go out and give you a wave as well. If you don't get your questions answered here, go ahead, throw them in Slido, and we'll be able to answer you next time we are streaming. We can pop on the intercoms at any time as well, so you'll be able to hear us.
Fantastic. Thank you.
That is awesome. And yes, I did pop in the chat there that you can access C2 via oceannetworks.ca. We also have a YouTube channel, which, as Tammy mentioned, will have some highlights on it. So you can check that out as well. And again, there is a link included in the chat as well to the entire expedition, where Tammy's talking about the Slido and the dancing with pool noodles. When you're out at sea, you gotta entertain yourself. We saw that lovely photo of the ship, and in the middle of nothing, what do you got around you? Blue in every direction. So let's make it fun.
I mean, we get creative on entertaining ourselves. I mean, there's a lot of stuff to do, but every now and then you still need a break.
Yep,
Sure.
Exactly. Let me see. There is a question actually here, and I'm curious if we can hear some multiple answers. The question is, are the deep seamounts insulated from climate change because of their depth? So maybe we can hear a little bit about that specifically, and then any other impacts of climate change we might see.
Anyone? I mean, I would guess they're maybe in the deeper parts, they could be a little bit, but overall, nothing's really protected from climate change. But maybe we'll get one of these experts in here to talk more about it. Yeah.
The ROV expert.
The ROV expert.
Yeah. Okay. The punchline is it's not good. So unfortunately, even though these seamounts are deep and remote, we know already that they are being impacted by climate change, and it's because of our previous expeditions and partnerships with long-term study programs like the Line P program at Fisheries and Oceans Canada. So the ocean water that bathes these very steep seamounts, maybe I'll start there. These seamounts are incredibly steep. Scone Kingless goes from being like a subtidal community, bang, down to like 3,000 meters, not very far away from that pinnacle. So they transition the water column really fast. The animals that live on the seamounts are fixed in space. They appreciate change, but on the order of geological time scales, so not very fast at all. But the ocean conditions in the water column are changing much faster. So in a study from our 2017 expedition in partnership with Line P, we know that we've actually lost 15% of the oxygen in our waters in the last 60 years. So it's 15% of the oxygen is gone. We already had some of the lowest oxygen water in the world, and now we've lost that. So it's called a severely hypoxic layer. It's also called an OMZ, an oxygen minimum zone, and it bathes these seamounts. The animals that live on them are experiencing these changes. And unfortunately, in combination with that and rising acidity, the outlook for the animals on the current trajectory is not good. It's something on the order of a lot of them will suffer local extinction within the next 100 years. So it's a somber thought, but I will leave you with, like, yes, climate change is impacting the deep sea, the world, everywhere. But with these seamounts, with these marine protected areas that we're working towards, we're trying to mitigate the things that are in our control. So the things that, like fishing, marine pollution, marine noise, things that we still can relieve these animals from, so that they're in a better situation to overcome, to adapt, to survive the oncoming changes, the changes they've already been experiencing. So yes, unfortunately, climate change does reach the deep sea. It is impacting our seamounts, and yeah, it's better to know, and it's thanks to expeditions like this with partners like we have on board that we know this work, and that we can get moving on the other protections that are in our powers.
Well, thank you for that response. So no escaping it, but we can do something to help.
I know. I don't know if anyone else wants to talk on such a somber subject. Nobody's jumping at the bit, but maybe Daniel, do you know of the like changes to the water community? No, Daniel is doing a little bit on time series work in the water column. So when you piece all our individual projects together, hopefully we'll get a more holistic view. But mine in particular is benthic, and so that's why I could speak to that. Yeah.
Awesome.
Oh, actually, Skiljada can speak about pelagic changes.
So I'm also a big zooplankton nerd. So we're in a really interesting area in this part of the Pacific, because we're right in between two a transition zone between two areas. We've got the Alaska current and we've got the California current. And what this means is that when the climate changes, that transition zone can shift a little bit north when things get warm, and it can shift a little bit south when things get cool. And so we have like La Nina and El Nino years, we have cooler years, and we have warmer years. We have a lot more marine heat waves, as well as heat waves on land. And what that means for the zooplankton is, in these warm years, we get smaller phytoplankton and we get smaller zooplankton, which is not as good fish food. And we also have copepods, which are little crustaceans. We have copepods from the south kind of dominating our area of the world. And what that means is that's not great fish food. They do not have a lot of lipids, omega-3 fatty acids, versus cooler years, we see a lot more big copepods, which is what we're bringing up in our bongos lately, which is really exciting. We're able to see these copepods with the naked eye. That's how big they are. Usually we need the microscope to see them. So those are the ones that are kind of rich in fatty acids, which means it's healthy for the fish. And we have good, healthy, thriving fish like all the ones we saw at Sahan Kingless. So I hope that helps. Northern copepods, cool waters, good for fish.
Awesome. Thank you. All right. We actually have a question in the chat question here. And again, we're doing our best to answer all of them. So if they don't go live, do check the writing, because we are going to type answers as well. But the question is, are there any potential studies on the evolution of life at hydrothermal vents? So maybe you did mention briefly that you're going to check them out. Can you tell us more about hydrothermal vents, even in general?
I'm not an expert in hydrothermal vents. I think they're really neat. They got me really excited about deep sea science when I was early on in days at UBC. The one we're going to check out tomorrow is an unconfirmed hydrothermal vent. It was observed a long time ago, and we want to know if it's still active. So this one's going to be an exciting find. In terms of the evolution and all that, I'll pull in someone else to kind of speak on that. Yeah.
Hi, everyone. Again, I don't know a whole lot about hydrothermal vents. And actually, this is a whole thing about why we partner with all of our different partners and colleagues and collaborators around Canada and the world. So this particular hydrothermal vent, we're working with scientists on the East Coast, and it's a partially unconfirmed hydrothermal vent. There was a survey in the night about 40 years ago, Sharice tells me, in this area. And since then, we've been collecting data for the last couple of years in the area, different maps and multi-beam. And now we're going to go out and see if we can actually find it using these old coordinates and a bunch of work that our colleagues on the East Coast have been working on to help us track it down. So we're going to learn a lot more tomorrow about hydrothermal vents. If we and let's wish us luck to actually find it.
Amazing that there's these entire worlds hidden beneath the waves.
Yes.
Very, very cool. I will just take a moment here, Tammy, and believe it or not, it is 3 minutes to 4. The time just flies past. And so we had actually scheduled this for approximately an hour. But do you have time to stick around a little bit longer if we have a few more questions?
Yeah, we've got another bit of time for sure. Yes.
Excellent. So folks, this is kind of that rallying call. We've got a few more minutes with these folks at sea. If you have any more questions, please do throw them at us. Maybe I'll throw you this one, Tammy, because it's a really neat one. You've been using terms like ancient. How do you know these creatures are ancient? How do you know how old a coral is or a rockfish is?
Oh boy, we got this question earlier this week, so I know the answer now. Okay, so corals they grow really, really slow. And so they grow just centimeters at a year. And so we actually were at Skunking Cliffs yesterday, and while I was in the control room, I asked the ROV pilots how high we were above the seafloor, and they told me we're 3.5 meters above the seafloor, and we were just missing the top of the coral. So this coral is 3.5 meters tall. So again, I needed some scale, and we asked my friend Chelsea how tall she was. So it's 1.5 Chelsea's tall, and we know they only grow like a centimeter a year. So we know that these are really, really, really old. So they're like old growth trees, thousands of years old. Rockfish, we have, there's all kinds of neat studies now, or over the years, we've learned that you can actually age rockfish by using their ear bones called otoliths. And they, again, I learned this week, can grow to over 200 years old. And so some of those old, huge rockfish we saw yesterday were likely over 200 years old. Anybody else?
That is absolutely amazing. Maybe you can tell us, so you've mentioned all of these amazing folks we've met, so many amazing folks. How did you end up in your role? Maybe we can get the quick version from everyone. What made you wake up and say, I am going to do deep sea expeditions? Or maybe you didn't.
Actually, I bet you a lot of us didn't know we were going to end up here. It's a really crazy road to how we end up here. Everybody's road is different. I started out as a young child just liking what was on the beach and knowing I wanted to be a beach biologist, really studying the beaches. In my role, it changes and grows into all kinds of opportunities to organize large collaborative surveys. So I've actually been on a deep sea ecology, a learning mission for about 5 years. So I'm just beginning to understand it. It's a huge and amazing ecosystem. So that's how I ended up here. But there's a lot of other really interesting stories and people are lining up actually to tell you them. So I'm going to pass you on to the next person. There you go. Thanks, everyone.
Hi, everyone. So I'm a bit earlier on in my journey to becoming a marine biologist, but right now I'm in grad school. So essentially how I started was I started in Ontario, which is a landlocked province. And I just always had an interest in ecology and how different organisms lived and everything like that. So Of course, I was much more familiar with the forests and with the lakes, but the ocean was always a big mystery and always super interesting to me. So just throughout school, throughout my classes, I would always just pick the opportunity that took me closer to the ocean, closer to studying marine animals and everything like that, until I finally made it out here for my grad studies at the University of Victoria. And then I got this opportunity to come out on this expedition, even though I am studying more the zooplankton and less to do with deep sea. So I really never could have imagined being able to be a part of this, but I made it out here and I'm loving it. Okay, here's Sheldon.
Hey guys, Shelton here. So I started working on boats at a young age. I was 19 years old and I've been working on boats for the last decade. And while I was working on the boats, I started picking up the camera, shooting drones underwater, all that kind of stuff, falling in love with the environment and the wildlife around. And that just transitioned into research and science and expeditions like this. So I've been doing this for about 4 or 5 years now. And yeah, I love it. And coming out on expeditions like this are always incredibly special.
Hi, everybody. So I guess my adventure started when I was about 4 or 5 and declared I wanted to be a whale veterinarian. I decided that wasn't going to work, but that I still wanted to study whales and learn about whales. And then I actually was introduced to hydrothermal vents. And sorry, Dad, the whales, they're still cool, but deep sea's cooler. And yeah, so it just grew from there. The more courses I took, the more I learned about all the itty bitty little stuff. The more excited I got about just the deep sea and the crazy life forms that live down there.
So like Lindsay, I did want to be a marine biologist when I was little, maybe not whale veterinarian, but marine biologist. So I did a degree in biology at UVic, and then I kind of went a different path to a diploma in environmental technology. During that, I did a co-op term, which I got with the Hydrographic Service, which led to working with fisheries and oceans. So I've just always made myself really available for new things. I have really pushed to get on a whole bunch of different surveys to really broaden my skill set, to make myself valuable to as many programs as possible. And then I've been lucky enough to lead the oceanographic program on the Deep Seas program since 2015, which is why I get to be involved with all these awesome people.
Okay, so for me, it was a pretty windy road to get to where I am. I was the kind of kid who loved playing in tidal pools, especially around Toe Hill on Haida Gwaii. I thought the ocean was really interesting. I didn't think I could actually be a scientist because it didn't feel like a real job. It was like, yeah, as an island girl, I'm like, there's no actual like scientists here that are like Haida and hold our perspective as well as like whatever it is that scientists do. So I thought I was going to be a doctor when I was in high school because I knew I liked the sciences, but I kind of clued in. I didn't, that wouldn't make me happy. I wanted to spend my time outside on boats, especially. So I just kind of like chased whatever interests me in school. And it just was ocean sciences, oceanography specifically. So not technically marine biology. I'm a marine biologist now, but I was an oceanographer, which is like more broad study of how the ocean all works in terms of all the different connections of the ocean rather than focus on just the animals. And I didn't think I would end up on deep sea expeditions, that's for sure. I didn't think I was going to go to grad school, but I wanted to keep learning, so I did. So when I was getting my master's, I saw the 2018 expedition that Jasal Juice was a part of, and I was just green with envy. I was watching the live stream when I should have been writing my thesis, and I was like, I definitely want to do that one day. So I did graduate. I did get a job with CHN, and I feel really lucky to be out here. And yeah, it's like, I don't know. I'm kind of a person who doubts myself every step of the way. It's like, we always say like, oh, anyone can accomplish anything, but I'm like, that's for like other people though, right? So yeah, it's pretty unbelievable, but you kind of just go even though you're scared, whether that's for going away to university or doing something new and challenging. like when Sharice really encouraged me to be the dive leader for the on Kingfisher dive, I was like, no way I can handle that. Like it's a lot to be the decision maker of like what happens on those dives and to make sure nothing goes wrong. But it was pretty spectacular, and I'm out on the other side of it. And when you do big stuff like that, you're like, I really can't accomplish anything. You just got to try the scary things and trust that it'll turn out well, and have really good mentors. So there's that.
All right. Like almost everyone else, I've taken quite a windy path to get to where I am. I've been in love with the ocean ever since I was a little kid. It was my first trip going to the ocean. I was like, I knew I wanted to do anything to do with the ocean. And I grew up in the US in Wisconsin. There's no ocean around. So that was quite a journey to get there. So I got a marine biology degree, thought I was going to work in a lab doing a lot of research. I particularly wanted to do invertebrates and biofuels. And then I ended up, after graduating with my marine biology degree, I taught an oceanography lab and went off to video school. And now I have a video degree. And I'm very lucky in the fact that my two passions of kind of these documentary style videos and just seeing this, sharing science with everyone, I get to combine both now. And as a video engineer that sails on research vessels, and our video gear mainly operates with ROVs, so I do a lot of deep sea. And so I kind of fell into what I do now, and I love it. I wouldn't change what I do. I still get to be my scientist when I want to, or help out, or still learn and still be on all that science side, but also going along with all the video side as well. But I had no desire to do any kind of live broadcast or anything, and that's pretty much what I do now. And so it's just be very, very willing to learn and change your plan, and don't be afraid to fail or try something new, because guarantee if you try it, you're not going to fail. So that's
Okay, I think I'm the last one in the line. And okay, so if the question is, how did you end up here? I ended up becoming the lead researcher for the deep sea ecology program at Fisheries and Oceans Canada. I'm going to try tell a different story than everyone else's, because so many of our stories are different, but they follow sometimes the same, like maybe it wasn't going to be us, or we didn't have the confidence, but it ended up working out and we're all here. But for me, my struggle was, I love the ocean. I was a surfer, grew up in South Africa, wanted to film the ocean, grew up watching Blue Planet 2. Just thought that was the neatest job, but I wasn't very good at school. And it turns out I was actually undiagnosed dyslexic. So I spent a fair bit of time failing at school and managed to flunk out of university the first time I tried. But I figured it out, and from the help and accepting help from other people, figured out that I was dyslexic. And I thought I would really, I need to give it a shot. I really want to work with the ocean. I want to have this adventurous life. I wanted it. So I went back to university, and those A's started to come, and then I met the right people, and oh my goodness, our coast is amazing. If you live on the west coast of Canada, and you leave this broadcast not knowing anything else, it's that our coastline is incredible, and it's full of all these deep sea wonders. And so I went to school at the University of Victoria, and I realized all the professors there were world-renowned deep sea biologists, and that through studying under them, I could attain this like adventurous ocean life. And I wanted it really bad. And now I look at my dyslexia as that quirky thing that makes me look at the ocean a little bit differently and maybe see patterns. And when I give talks to younger kids, I just think, whatever that thing that makes you different, whether it's coming at it from videography or where you grew up and your culture's connection with the environment around you, that thing about you can be your superpower, and it can be the thing that gets you that extra step that makes it that you get to sail the deep sea with cool people and chat to people around the world about how rad the deep sea is. So yeah, that's how I got here, and I wouldn't wish to be anywhere else. Yeah.
Wonderful.
Pass it back to you, Monica.
Thank you, everyone, for those wonderful answers and positive words of wisdom that our audience can bring back home with them. We've got a question which I love. Do you ever take volunteers out at sea with you?
That's a great question. So the short answer is, well, the boat's only so big, and there's a lot of people who want to be out here, and that's why we have so many partners that are remotely piping into every dive. So unfortunately, we don't just get to take volunteers. Monica, I feel like that question was from you, specifically about you.
It actually wasn't, but I am guilty of feeling that way.
Yeah. So we prioritize representing the co-governing nations. We prioritize multidisciplinary science. We prioritize training the next generation of scientists. So grad students, representation, and the oceanographers, the benthic ecologists, the technicians, the pilots. And so by the time you finish with that list, it's a little bit packed and cramped on here already. So I'm sorry to say that it doesn't usually go out to the public for volunteers. But if you watched us nonstop, you could feel like you are out here.
Sounds like we just need to put more money into ocean science so we can have bigger boats.
Bigger boats.
Bigger boats.
Yeah.
Excellent. No, it is a great question for sure. And actually, this is related to this. We have another question. Were there any artists involved in the expeditions this year? Or will there be in the future?
Can someone please bring cups?
Oh, the artists.
Oh, maybe our ship. Our ship. Oh, can we bring our ship? Fantastic. So one of the positions we have on board is our filmmaker, photographer, Shelton. So that's making digital assets. And Tammy, thank you. So we have photographers on board that are making digital assets, and it's art. It's creating videos and photos that we share with everyone. So if you've been on social media, you know how much we're trying to get the information out there. So we have 2 people on board. At least one of them has 2 jobs, and creating that content is one of them. But amazingly, unfortunately, some of the Canadian Coast Guard, which we work very closely with, we create this art, this map, and we had these drawn by the Canadian Coast Guard. So I'm going to try get them. So these are magnets of the telly, that we have, and we put on our map and we drive them around. So we got this art going on. This is spare time art by Tammy G. So what is it, knitting? It's not knitting, but it's crocheting, little Dumbo octopus. And then there was another one. Oh, Josh was out here in 2019 from the Nuu-chah-nulth Nation. I have them here. They're a partner, and Josh, in addition to studying oceanography at the University of Victoria, Josh is a phenomenal artist, and he was here to capture the experience in part, and he produced art that represented the animals that we saw, and that was a really phenomenal experience. So we bring videographers, photographers, we all dabble in little arts in our spare time, and then artists. We'll end that question there.
Oh, sounds like it, for people who didn't believe we really were at sea, you really are at sea. Sounds like we've got some announcements and things like that. And actually, maybe we can wrap up with this last question here. What's your favorite part about being at sea, either on the ship or in terms of discovery? What's your favorite part in a soundbite? I know that's a tough question.
Can we all do one soundbite? Can we do one sentence soundbites? We'll just cycle through so fast. I promise we'll like push each other out. Okay. My favorite part of being out at sea is that I walk out of this lab and I am surrounded by the ocean, and it is so inspiring.
All the different people I get to meet and scientists and their excitement for different things that we are doing.
Same as Tammy, we get to meet some amazing people and we get to learn tons of new things, and it makes it all worthwhile.
For me, it's the variety of every day. Every day is a little bit different, and you never know what tomorrow is going to hold.
I love to be out here away from land and just humbled by the power of the ocean.
Power of the ocean. It's the amazing people and the challenge of being able to pull this all off and work all together to make it happen.
The creatures and the habitats that I get to see for the first time are as close to as in person as I can be.
The most special part to me is visiting on Kinkless and acknowledging the beauty and the connections we have to that sacred place.
And I would say for me, one is the amazing wildlife that we see. We saw some Dall's porpoises swimming past, and also the fact that the ship rocks you to sleep at night. It's quite comforting.
Okay, let's all pack in there. Also by the other. All right, we're getting in.
Say bye.
All right, thank you. Yes, this is a fantastic way to wrap up. Look at all these. Again, you rarely see a picture so smart. Look at all the brains you have there. So thank you everyone who participated. If your question didn't get answered live, oh, there we go. We were fading out there, but we're good now. If it didn't get answered live, we will type an answer for you in the chat. And again, coming up in the chat, watch for all those links. We have got those wonderful links you can watch online at the expedition. You can ask questions in the Slido, all this kind of good stuff. And Ship, I will save the chat for you because we have just amazing thank yous, wonderful responses to your stories, safe sailings. There's so much here. And again, it would take another hour to go through, but I'll save it for you. And we just can't thank you enough. So thank you, everyone's, and wonderful.
One question from us.
Oh,
One
yes.
question. How do we get a copy of the recording?
Copy of the recording? I don't know. How do I get on the ship?
Shots fired.
No. We will do our best to make the recordings available on the Ocean Networks Canada YouTube after this. So again, you can head there to check it out. And we look forward to seeing your questions coming in still on the Slido. We look forward to you visiting all those resources, and yeah, a little shine and polish on this, and then it should be available on the YouTube of Ocean Networks Canada YouTube.
Awesome.
Cool.
Great. Thank you.
Thanks, everyone.
Cheers.
Bye.
Awesome work, team. You did great again.